Yeah.
So good morning and good afternoon to esteemed colleagues, professors, deans, students, and distinguished guests. It is with great pleasure that we welcome you all today in an exciting webinar titled From Big Data to Precision Medicine, Leveraging Artificial Intelligence and Bioinformatics in Cancer Research and Therapy. This event is organized by the Faculty of Medicine, the Advanced Research Center, and the Health Service Policy Application and Research Center of Istanbul Aydın University. We are honored to have such a distinguished audience join us for that important academic gathering. This webinar marks the second seminar in our Artificial Intelligence series, where we continue to explore the profound implication of artificial intelligence in healthcare. As researchers, we are well aware of growing importance of interdisciplinary approaches to tackling some of the most pressing challenges in medicine, particularly in cancer research and therapy. So today we are privileged to have Dr. Peng, a distinguished faculty member at the prestigious National Institute of Health at the National Cancer Institute, NCI, as our guest speaker. So Dr. Peng is a highly respected scientist known for his groundbreaking work in bioinformatics and precision medicine, and especially his significant contribution to advancing our understanding of how artificial intelligence can be leveraged to improve patient outcomes, particularly in the area of cancer treatment. So we are eager to hear his insight into the role of artificial intelligence in cancer research and therapy. But before Dr. Peng begins his presentation, I will briefly introduce his impressive academic background and his invaluable contribution to his field. So we can continue with where Dr. Peng completed his PhD at the Department of Computer Science, Lewis Sigler Genomics Institute of Princeton University, where he specialized in bioinformatics and computational biology. Prior to this, he earned his undergraduate degree with the highest nation honors from the Department of Computer Science at Tsinghua University, where he ranked first in his class. So his academic journey reflects a remarkable commitment to excellence, and his extensive expertise in the application of computational methods to biological research has positioned him as a leader in his field. So when we continue with academic career, Dr. Peng started his research program at the National Cancer Institute in 2019. His lab focuses on developing big data and artificial intelligence frameworks to identify biomarkers and new therapeutic approaches for cancer immunotherapies in solid tumors. Before joining NCI, he completed his postdoctoral training at the Dana-Farber Cancer Institute and Harvard University. During his postdoctoral research, Peng developed computational frameworks that repurpose public domain data to identify biomarkers and regulators of cancer immunotherapy resistance. So notably, his computational models revealed that cancer cells could utilize the self-production strategy of cytotoxic lymphocytes to resist lymphocyte killing under immune checkpoint blockade. So when we continue with academic duties, we see that Dr. Peng has served as a stand-man investigator at the Cancer Data Science Laboratory and as the head of computational cancer immunology section at the National Cancer Institute at NIH. So in these roles, he leads cutting-edge research in computational cancer immunology and plays a pivotal role in advancing the use of big data and artificial intelligence in cancer research and therapy. And of course, when we check for the research facility, we see that in research area, he's focused on developing integrative frameworks that leverage big data resources from public domains to identify key regulators of cancer therapy resistance. And one of the major challenges in cancer research is the limited data available to fully, in fact, understand the clinical efficacy of treatment. So especially as new drugs with distinct mechanisms of action are approved each year. So to address these, Dr. JIANG team is developing advanced statistical and machine learning infrastructure that transfer knowledge from large existing data cores to explore new cancer biology problems and improve therapeutic strategies. So his area of expertise include data science and computation biology with a particular focus on big data integration and cancer genomics. Additionally, Dr. Peng utilized machine learning and biostatistics to drive advances in precision medicine, enabling more effective personalized treatment options for cancer patients. And of course, he graduated from Princeton, we know that, with his undergraduate studies with the highest national honors at the Department of the Computer Science and Tsinghua University. And of course, he's a recipient of several prestigious awards, including the NCI K99 Pathway to Independence Award, the Scholar in Training Award from the American Association of Cancer Research, and the Technology Innovation Award from the Cancer Research Institute. So these honors reflects his outstanding contribution to cancer research and his significant impact in the field of computational biology and cancer immunotherapy. So when we continue, we see that his research has earned him a high reputation with over 9,000 citations and widely recognized and have greatly influenced the advancement of computational biology. So at the end, we can continue. And we would like to thank you for all being us today. We would like to thank Dr. Peng JIANG for sharing his invaluable knowledge and expertise with us today. So we are truly grateful for his time and insight and look forward to a highly enriching session filled with insightful discussion and academic exchange. So now, without further ado, the stage is Dr. Peng's, and I make you as a co-host now, and you can share your presentation with our students and academicians that are here. So thank you again.
Yeah, so Arta, can you see my screen right now?
Yes, we can see, and it is already full screen. Yeah, thank you.
Perfect. Thank you so much for the fantastic introduction, Arta. And everyone, I really apologize again for being late. Because the US shifted from the summertime to winter time, I forgot to move my calendar like a backward for 1 hour earlier. So just sorry again. Okay. So today I'm going to talk about some of our recent attempts. I would say it's a recent tries on using artificial intelligence in translational cancer research. The result might be a little bit disappointing for many people here, as I will show. However, I do think that our setbacks and a few recent failures in using artificial intelligence are very essential for everyone here to know what is the limitations of artificial intelligence nowadays, and also what is the correct expectations of AI. So in the first part of the work today, I'm going to talk about our recent tries on using AI to design small molecule binders for FIBP protein, and I will talk about why we are interested in this project. So as everyone say in the public media, we are in the age of breakthrough by artificial intelligence, as evidenced by the stock price of Nvidia. It's really crazy. And also the Nobel Prize in this year were given to artificial intelligence because they have solved like one biggest problem in biology for many hundreds of years, protein folding. But whether this is true, let's see, okay? So, and also recently, the AlphaFold 3 team, which is a Nobel Prize winning team, claimed that they can predict the interactions of all life's molecules. So it seems that with artificial intelligence, there's already a big, huge breakthrough of molecular biology. But is this really true? Let's look at the Nobel Prize winning work this year. AlphaFold. So they claim that they can resolve the protein folding problem. This is why they got Nobel Prize. However, at the National Cancer Institute, our focus is really different from a pure computational perspective. We really care about biology. Is this really true biologically? So in cancer, there are plenty of examples of one single mutation will damage the protein function. We call it like somehow like an oncogenic mutation on tumor suppressors. Okay, so in these two examples, in the first example, UBA1 protein, there is a mutation on IL-198A, this single mutation will damage the hydrophobic pocket of this protein. Of course, if you damage the hydrophobic pocket of the protein, it will damage the entire structure. But when you input the UBA1 IL-198A mutation into AlphaFold, it will predict a protein structure that's almost the same as a wild-type structure, which means AlphaFold does not really predict the structure of this mutant protein. This makes sense because fundamentally, artificial intelligence nowadays by deep learning is just a nonlinear regression. By nonlinear regression, if you just change one position, it will think this is exactly the same, very similar input, and then predict the same structure. And in the second example, the BRCT protein is the same. One single mutation will be sufficient to change the protein structure on this position. And if you input this mutant protein, AlphaFold will predict exactly the same, almost the same protein structure. If you align AlphaFold prediction and the real crystal structure determined, the real structure, you will see they are pretty different. AlphaFold did not really capture the true mutant protein structure. So in short, it's just too early for AlphaFold to claim they have resolved the problem. For all of the important mutant protein structures in cancer research field, AlphaFold does not really predict that, okay? And this really makes sense because nowadays AI is just nonlinear regression. It's still far away from really solve this problem. And then the second question is more relevant to our to the talk today is, can AI design new drugs? Because one important reason for AlphaFold to get a Nobel Prize is because it can design new drugs, or at least make new drug design much easier and faster. But is this the truth here? If you open the AlphaFold 3 software and to predict a ligand and protein interaction, what you will see? They allow you to input any protein sequence. However, remember, protein structures are determined by both protein sequence and also post-translational modification, such as phosphorylation and this kind of modification. But AlphaFold completely ignore that. But it's okay. But then once you input the ligand, AlphaFold 3 only allow you to select a handful of molecules, such as ATP, ADP, GTP, which are small ligands, have plenty of structures, crystal structure in the PDB database. It does not allow you to input any small molecule sequence or structure. Therefore, it definitely overclaim. It has not really resolved the problem of all life molecules interaction. It's just from the paper title. In the real software, they does not allow you to input that. They can only predict ATP, GTP, this kind of common small ligands binding with proteins. But fortunately, there are some companies, their software allow us to try any kind of small molecule and protein structures. There is a kind of a startup company in the US. They claim the breakthrough of AI technologies in drug discovery called MoreSoft. And lots of large pharmaceutical companies are their partners, such as Lilly, Pfizer, Sanofi, Novartis, Bayer, and also including NIH. NIH eventually fund the money to pay them. It's a really expensive software license to just buy their software and try small molecule designs. Okay, let's see whether this AI technology really works. So in 2022, my group published this paper on finding a new drug targeting T cells. We first established a computational model called the tumor resilient T cell model to identify the molecular characteristics of T cells that are resilient to immune suppression in solid tumors. The basic idea underlying the T-RAS model is that we look at the T cells, what kind of molecular signals they receive. For example, does T cell receive lots of TGF-beta signal? If yes, we see, okay, this T cell is under immune suppression. Otherwise, we see it's not under immune suppression. Then we look at the T cells that receive lots of immune suppressive signals and see that which T cells are still proliferative, even they receive lots of immune suppressive signals. Then we analyze their molecular characteristics, and we found there's a gene called FIBP as the #1 negative markers, which means T cells with high FIBP gene expression tends to be really sensitive to immune suppression in solid tumors. Then we knock out the FIBP in T cells and perform the adoptive T cell transfer into mouse. We see that the FIBP knockout T cells can achieve a very efficient inhibition effect on mouse tumor growth. And after this publication, we have been searching for a FIBP small compound inhibitor because adoptive T cell transfer is not a really widely applicable therapeutic approach. You need a very dedicated manufacturer facility to make the adoptive infusion product. Not many hospitals in the world have this facility. Even in US, only a handful of hospitals can make this kind of a therapeutic product. So first, we further validate our findings using human donor T cells because previously our experiment is in mouse, right? Like we use the plasmid from our NCI colleagues who are trying this GPC3 CAR-T in their clinical trials. We knock out FIBP, and in the GPC3 CAR-T, we see, okay, indeed, as we expected, FIBP knockout can significantly enhance the GPC3 CAR-T killing effect in our in vitro assays. This further makes us confident about FIBP as a good drug target in T cells. Before searching a small molecule inhibitor, we want to say that currently all AI algorithms predict a binder. A binder is not an inhibitor, and also an inhibitor is not equivalent to a drug because its pharmacokinetic properties may not be really good. However, we can only start from searching a binder as an initial step. Okay. So AlphaFold gave a very high confidence prediction on FIBP structure. There is no previous molecular structure determined for FIBP. You can see that AlphaFold gave a blue score, which is very high confidence on most locations, and also predict a few disordered region on the FIBP protein. And then we run the AI algorithm NIH purchased from MoreSoft software. The software run like this. First of all, the software will identify potential binding pocket. On the AlphaFold determined structure of FIBP, the MoreSoft software identified 4 potential pockets. And then they perform virtual docking using artificial intelligence to test, like out of several million out-of-the-shelf small molecules, which of them can bind to any of these small docking pocket. And finally, the software gave us 10 high confidence prediction, which means the software are very confident about the reliability of the small molecule binders they predict. Initially, when we look at this top 10 prediction, we are very happy because, first of all, these small molecules have some chemical themes, which means they are not some random small molecule. They do share some common molecular themes. As you can see, there are some benzene rings, and also there are some polar groups, which means they should be soluble in water. And then we validate whether this top 10 prediction can bind the FIBP protein. Our validation approach is based on technology called microscale thermophoresis. The microscale thermophoresis is testing the moving speed of the protein labeled. If the protein is unlabeled, you shoot the protein with a laser, they will move away from the center. However, if the protein is bounded by a small molecule, the moving speed will change. Could be faster, could be slower, depending on the property of the small molecules that bind to the protein. So on the equipment, what you determined is the shape of the signal curves. Once the protein is bound, the curve should shift from the unbound curve. Okay, this is a basic idea. So we paid some companies to synthesize the FIBP protein, label them, and then perform the experiment one by one. So first of all, the MST curves, the microscale thermophoresis curves, like some of them are really wild. Kind of, they are not forming the smooth curves. This means there are some aggregates. So once we paid some chemical factories to synthesize these small molecules one by one. So because once AI predict some molecule bind to a protein, you still need to pay some chemical compound companies to synthesize them. The synthesization process could be expensive. And then after that, we found lots of them, actually 6 of them formed aggregates in solutions, which means just from the structure, you think they are soluble in water, not necessarily. They could still form aggregates, either covalent aggregates or non-covalent aggregates by unknown reasons. And only 3, only one of them somehow have a good water solubility. By the way, 3 of the top 10 predictions cannot be synthesized, which means the chemical synthesis company provider has tried their best, but still they cannot really synthesize this compound. And then once we run the MST assay, you can see that the overall shape is very marginal. This is the unbound protein, this bound protein, they are very close to the unbound curve, which means there's no binding at all. So in short, we paid tens of thousands of dollars for synthesizing this compound. Not all of them can be synthesized after AI predictions, and 6 of them do not really have perfect water solubility, and only one of them have good solubility, but then all of them does not really bind to the FIBP protein, even if they are top 10 prediction with very high confidence by AI. So in short, at least according to our evaluation about the drug target we identified, artificial intelligence does not give any meaningful predictions on small molecule binders. And by the way, AlphaFold, by our evaluation on mutant protein in cancer, does not give the correct prediction. So right now, AI is still far away from being able to help on drug design. We need to be very cautious when we are working on this in this field. And in 2022, our colleagues at the Cancer Data Science Department write this review paper together on Nature Reviews Cancer. And then, in this review, we discussed a case report by FDA, Food Drug Administration of USA, and the FDA report mentioned one thing. So nowadays, there are many AI works claim that focus on digital pathology, like imaging data, not small molecule design. So they look at digital pathology data, they use AI to prove that they are doing better than clinicians, okay? However, the FDA report challenged the study design of this algorithm, because the way their study designed is to look at only one digital pathology image, H10E staining, most of the time. And then they compared AI performance with pathology performance. However, this is really unfair comparison because clinicians never make decisions by one single digital pathology image. Instead, clinicians could get multiple biopsies. If clinicians is not confident about the diagnosis, they could order a immunohistochemistry staining of a few cancer cell markers. So for example, recently, one of my best students before, unfortunately, was diagnosed with cancer, breast cancer. And I asked her, she told me that her diagnosis was definitely not based on H&E, but based on immunohistochemistry on a few common breast cancer markers like ER, PR, HER2, and also based on a bunch of other evidence. So which means the clinical decision system is based on multiple biopsy, it's based on immunohistochemistry, and also based on multiple other clinical factors. But the AI studies only compare human performance on one single H&E image. It's not comparing against the whole clinical decision system. Therefore, the FDA report says that even though they approve this kind of AI diagnosis as a device they can try in clinical settings, but it's still premature to claim AI are doing better than the current clinical system because the performance comparison is highly biased. And recently I just see some Nature Medicine paper finally performed the performance of AI versus the clinical decision system based on multiple biopsies, based on IHC, and their conclusions AI is doing significantly worse. So in summary, even though people are feeling super excited about AI, and also at least in US, venture capitalists will not give any money if you don't claim you are an AI company. But in reality, our AI nowadays is only doing reasonable job on some small task. But in terms of the drug design, clinical decision is far from being usable at all. But maybe our evaluation is highly biased, but at least this is according to the evaluation by me and our NCI colleagues. We need to be very cautious on, well, everyone is being so crazy about AI nowadays. Okay. And in the second part of the talk, I'm going to introduce a paper we published last year on as a computational framework on analyzing spatial transcriptomic data. Many of my colleagues from the wet lab and the immunology part are finding spatial transcriptomics are very useful in their research. And I believe maybe many labs in Turkey are also using this spatial transcriptomic data. And we designed this SpaceSet framework specifically to address a few challenges in the spatial transcriptomic analysis for tumor data. This overall framework for SpaceSet, for most spatial transcriptomic data, they don't really give you single cell resolution data. Instead, each detection spot makes the signal from about 3 to 10 cells. Because if you use a single cell spatial transcriptomic data, you will not really get a genome-wide coverage. So the step one will be decomposing the cell fractions in each single spatial detection spot. And also a common downstream analysis is to identify the ligand receptor interactions from spatial transcriptomic. There are many computational frameworks published for this analysis task. But why do we still design one more computational framework? Here's the reason. So previously, frameworks are not really good at addressing a few key limitations in the spatial transcriptomic analysis for tumor samples. Tumors are very heterogeneous across different regions across patients. Typically, we don't have a matched transcriptomic profile for malignant cell for each case. Therefore, it is hard to decompose the tumor cell fraction using most previous computational softwares. And second, many immune cell types, their expression signatures are highly closely related, which means they will close high collinear relative when you are decomposing cell fractions. This high collinear profile will create lots of result variations in the result of cell lineage fraction decomposition. And also, in the spatial transcriptomic data, the cell density will vary significantly across different tumor regions. Most previous computational softwares only decompose all cell fractions and add up them to one, but this is wrong. Some regions will have more cells, some regions will have less cells. Therefore, we need very dedicated computational solutions to address them. So first of all, we look at how can we reliably estimate the cancer cell fraction as each detection spot, even without any malignant cell signature for each case specifically. So in bioinformatics field, what people commonly do is using a software called InfoCNV. CNV means copy number variation, because there is no universal there's no universal signature for cancer cell. However, cancer cell tends to have a large-scale copy number variation across different genome regions. The InfoCNV software aggregates all of the spots together and tells you which spot has lots of copy number variations informed by gene expression. They just look at across the different chromosome arms, whether some chromosome arms have a large-scale amplification or deletion. However, this InfoCNV software, even though they are widely used in bioinformatics field to infer cancer cell fractions, is actually fundamentally wrong here, or at least you need to use it very carefully. Because the InfoCNV procedure needs you to select a bunch of reference cells. If you select, unfortunately, lots of immune cells, including lots of macrophage, you are going to get lots of illusions, because in macrophage or in general, any antigen-presenting cells, the HLA gene complex will be highly amplified on chromosome 6 arm. And then if you use these cells, you think they are normal cells, they are not malignant cells, you use them as a reference, you are going to get an illusion of a chromosome 6 deletion and classify lots of normal cells as cancer cells, but this is actually wrong. But then if you use some cells, such as stromal cells, as the reference profile, you don't have this problem, but you are going to get a lot of cells with chromosome 6 amplification. In short, because of cell lineage, lots of immune cells still have a large-scale chromosome arm overexpression, and the InfoCNV will think that, okay, they are cancer cells, but they are actually not. This is the first challenge. And second challenge, many tumor cells don't have copy number variations. This study from the TCGA consortium showed that the cancer cells can roughly be classified into high copy number cancer cells and high mutation cancer cells. In the high mutation cancer cells, you don't see copy number variation. Therefore, the InfoCNV is not really a reliable approach to infer cancer cell fractions for single cell or spatial transcriptomics in general. So this is our solution. The Cancer Genome Atlas project has used the SNP array to identify the copy number variation patterns for diverse human cancers. Currently, SNP arrays are considered as the gold standard for copy number variation identification. And then you can see that in each cancer type, the SNP array patterns are kind of consistent. So for example, breast cancer, you typically see this chromosome 1p arm and chromosome 8 arm amplification in most of the tumor samples. And the same is true for glioblastoma. You see the chromosome 7 amplification and chromosome 10 deletion in most samples. Therefore, we use the SNP array patterns as a dictionary and create a bunch of cancer cell copy number variation dictionaries to identify whether some spots have similar patterns. So here's our procedure. Once we have a spatial transcriptomic, we look at whether the detection spot transcriptomic profile are similar to one of our copy number variation dictionary. If yes, we will infer a cancer cell fraction there. But if no, we will look at another dictionary, which is the tumor normal expression dictionary from the Cancer Genome Atlas, and infer the cancer cell fraction there. And overall, based on our simulation study, our copy number variation, our expression dictionary performance are very close, and they are all doing pretty good job on identifying the cancer cell fractions. And the second challenge of deconvolving cell fractions at each detection spot, as I mentioned, is the high similarity between the immune cell lineage. So here is a maybe more intuitive way to show that. For example, like a lot of times in our bioinformatics field, people try to deconvolve these subtle lineage fractions like T helper 1, T helper 2, T helper 17. But in reality, their expression profiles are highly similar to each other. If you run a linear regression with all of them together, the collinearity of the covariance will make the result highly unstable. So our solution is to use the hierarchical regression. In the level 1 regression, we only aim to identify the fractions of cell lineage that are reasonably different from each other. And in the level 2 regression, we freeze the coefficient in the level 1 regression result and run the optimization of the coefficient on the level 2 without affecting the level 1 regression coefficient. As you can see, according to our different evaluation metrics, our constrained hierarchical linear regression significantly outperform the regression with all covariants together. And the third challenge is about the cell density. So here is a more intuitive example. In this breast tumor spatial transcriptomic profiles, there are some regions with very high cell density, but there are also some regions with very low cell density. If you deconvolve cell fractions and add up them to 1 at every location, this is unfair for the high density cell regions. And our solution is to import to put an unknown cell fraction component in the regression procedure. If in the low cell density region, typically you will see a very low UMI count. UMI means unique molecular identifier, which means if you just count the unique molecular identifier count from the data, you will see that the low tissue density region will have a significantly lower UMI. And then this low UMI will trigger a high unknown cell fraction in the linear regression, therefore down-tune the overall cell fraction. This is a way we can automatically adjust the cell fraction and make cell fraction much lower in the low cell density region. So then we use the human pathology labelings to evaluate the accuracy of our procedure. So we ask our pathology collaborators to annotate the matched histopathology stats for each spatial transcriptomic data into 4 regions: tumor region, stromal region, and the lymphocyte region, and the macrophage region by the signal called hemocyte deposition. In general, it's very hard to identify macrophage region from H&E image. But if you see some signals of some markers of hemocyte deposition, you can see, okay, there might be some macrophage there. And then we compared our SpaceSet framework against 10 other previous softwares using our human double-blinded annotated region, you can see that SpaceX outperformed all other computational approach on different tumor regions. And then we further develop another component of SpaceX on studying the cell-cell interactions, because one key applications of using spatial transcriptomics is to study cell-cell interactions. In the current bioinformatics field, there are two typical broad ways to infer cellular interactions using spatial transcriptomic or single-cell data. The first category is you get a transcriptomic profile, you infer which signals the cell receive, either bulk tissues or single-cell tissue. The second broad category is giving a spatial transcriptomic data. You infer the ligand receptor interactions in the spatial transcriptomic data. So today we will talk about the second category, but I will briefly introduce some of our recent comparisons of a bunch of softwares in the first category, which is given a gene expression profile. Can you infer which signal the sample receives? Recently, there's a Nature paper published, they inject different cytokines into the mouse, and then perform single-cell RNA sequencing after cytokine injection into the mouse, and then they establish an immune dictionary. With a gene expression signature from the immune dictionary, you can infer which cytokine signals your sample receive. And another popular approach is called the niche net. They use the large amount of protein-protein interaction data to construct a network. And then they look at for each cytokine ligand, what genes are there in their in their protein interaction network, and use this gene set to infer which cytokine ligand your samples potentially receive. And the third popular approach is one approach that I developed several years ago in 2021. It's called CytoSeq, which we collect more than 10,000 cytokine treatment response profiles in different cellular models from the NCBI GEO and the ArrayExpress database. And using these cytokine treatment response profiles on different cellular models, we can infer which cytokine response signals your sample receive. We performed a cytokine response prediction quality evaluation using a gold standard from human anti-cytokine therapy data, because in our opinion, only human clinical data from anti-cytokine therapies from human inflammatory studies are gold standard to evaluate whether software can really capture the cytokine effect. In this kind of studies, patients are treated by anti-cytokine therapies. For example, this one is the anti-IL-1 neutralizing therapy among a psoriasis patient. And then after they treat the patient, they get the peripheral blood gene expression profiles on day 3. And then on day 15, they measured the clinical response, either 100% response or 0 response or placebo. And then we used the different softwares to predict their IL-1 beta cytokine activity on day 3 and see whether day 3 can predict the day 15 final clinical response. And you can see that our CytoSeq platform reliably predicted the final clinical response. This is the approach we use to evaluate the cytokine activity prediction. And overall, these 3 popular approaches, NicheNet have the largest number of ligands included, because of course they use the protein-protein interaction networks, has lots of proteins included. However, in terms of the capability on predicting cytokine inhibition from anti-inflammatory clinical studies, NicheNet and all the other softwares are not doing so well, and cytosine is top performer. The recent Nature paper on immune dictionary performed really close to random. Initially, we are very concerned about this, whether we have done something wrong in the evaluation procedure. But after we double check, we found the potential reason. Because when people generate the data from the immune dictionary, they use protein from E. coli. As we know, cytokine proteins or any proteins produced from E. coli has a lot of endotoxin included with the protein sample. Once you inject the protein into the mouse, the endotoxin in the protein samples will trigger a LPS response pattern. Therefore, you are going to get a highly confounded cytokine response profile by using E. coli produced proteins. And also, inject cytokines into the mouse will trigger panocytosis. Most of protein will disappear after 30 minutes, which probably explains the low performance of the immune dictionary here. Okay, and then we jump to our SpaceX framework to study, to see whether the SpaceX can identify the ligand receptor interactions in spatial transcriptomic data. And here is an approach. We first look at the co-localization between two cell types. Here is the example between cancer social fibroblast and M2 macrophage. You can see that the blue spot are M2 macrophage, the red spot are cancer-associated fibroblasts, and they are co-localizing on the specific region. And then we look at, for the co-localization regions, whether they are enriched for the co-expression of some known pairs of ligand receptors. We see the co-expression of the known pairs, and we shuffle this protein in bipartite networks. We see whether the shuffled pairs have lower expression in general, and we only predict a ligand receptor interactions if there's a cellular co-localization, and also there is a co-enrichment of the ligand receptor expression levels. Interestingly, we found that all of the cell-cell interactions and the ligand receptor interactions are enriched on the spot close to tumor immune boundaries. Here are the tumor cells, here are immune cells, the black spot are the boundaries. All of our interactions we found are highly close to the boundaries. And then when you analyze the tumor cell gene expression close to the interaction spot and far from the interaction spot, you can see a very interesting phenomenon. Tumor cells far away from the ligand receptor interaction high spot are enriched in high proliferation signatures. But tumor cells are close to the immune cell interaction spot are growing slower, but highly enriched in the epithelial mesenchymal transition signatures, which means these cells are highly aggressive, even though they grow slower. This is the overall summary of the second part of the talk. Like we developed this SpaceX framework for cell lineage deconvolution for spatial transcriptomic data and inform intercellular interactions based on ligand receptor interactions. This is our source code, and this is our paper. And thank you so much for your attention. And last of all, I will give sincere acknowledgement to my team members and to my alumni, and also to colleagues and mentors at NCI. Without their help and hard work, nothing is possible here. Thank you very much for your attention.
Dear Dr. Peng, thank you very much for such a grateful presentation, and it was full of knowledge for us and for all of the students here.
Thank you, Arthur.
Of course, we have a lot of students that are in their conference hall that are watching you from there, and they will have some questions, if it's okay for you, if you...
Yes.
So I would like to ask now the students and academicians that are here, if you have any question, you can write in the chat, or you can ask directly here. Yes, I'm waiting for the question.
Yeah.
Or...
Well,
there are students here that can ask directly if they increase In fact, I had a question which was related with your tide model, computational model, but already you explained everything in the presentation, because you mentioned that how the artificial intelligence should be, in fact, should be compared against the clinical decision. So it was it has a very high impact in the research. So already we have one of our guests is Professor Zarghami. If you would like, would you like to ask something?
Oh, hi, Arta. Yeah. So you have probably mute.
Dear Professor Zarghami, could you please open the voice, because we cannot hear you?
Right now
Yes.
is okay, Dr. Arta?
Yeah.
I really appreciate Professor Peng for nice presentation, especially give all new knowledge, in fact, happening in the area of the cancer research, especially using AI system. My question is that, but I didn't know that any information about the survival analysis, so all the interaction molecules inside the cell-cell interaction or molecule-molecule interaction inside the cells, especially for the in fact the survival analysis of those interactions happens in clinical trial or after research. But I don't know, maybe Professor Peng, any idea?
So so first of all, could you teach me how to pronounce your name, like Zarghami?
Yeah, Zarghami. Yeah, my name is Zarghami, Zarghami.
Yeah. So Professor Zarghami, thank you for your question. Let me try to recap your question. Are you asking that whether we have been testing the relationship between cell-cell interactions and patient clinical outcomes in our study?
Yeah, that's true. That's my question.
Yeah. Actually, Zarghami, this is the key question we are studying right now. Because I completely agree with you that the association with clinical outcome is the most important aspect of cell-cell interaction, right? Otherwise, why do we really study them? We have an ongoing study, not published yet, still ongoing. We studied the secreted protein and downstream cell interactions, and we identify a signature of a secreted protein downstream cell interaction and use this signature to identify whether the secreted protein cell interactions are associated with patient clinical outcome in the large amount of patient cohort we have collected from immunotherapy clinical trials. And we do identify a few secreted proteins that people have no knowledge about their role in cancer before, but their interactions, their intercellular interactions are highly associated with either bad or good immunotherapy outcomes. And then we further validate them in mouse. We synthesize the protein and indeed find one of the protein is a really promising good immunotherapy target. So I have not included that part in my talk yet because this product is really in the middle. We have not really published yet, but indeed this is a association between intercellular interaction and clinical outcome is a key focus of my lab right now, and we think we find some promising immunotherapy target. And hopefully, once our paper published, I will contact you again, Arta, to present our new works.
Well, that's interesting. Is it possible to ask another question, Dr. Arta, Professor Peng?
Yeah, of course.
Yeah, we know that in fact it's the real fact about the system, especially in cancer research or cancer cells or in the chronological aging systems. So we know that by the time all the cells is going to become aged, by the way producing some secretomes, in fact they stimulate the immune cells. For example, macrophages or some. These macrophages also produce some inflammatory factors. So yeah, that's the point. Right now, I'm really interested to look at some metabolites inside the cells, based on how come we can manipulate or at least inhibit this kind of proteins, especially the enzyme proteins, in fact, the degrading or the power of energy of the cells. So that by that, I don't know, is any possibility to look at by AI systems or AI, in fact, softwares, or look at the inside and find some metabolites. How come they can manipulate or modulate this process of the aging systems, especially in cancer or cancer looks like a rejuvenation. Exactly, the cells back to the rejuvenate.
Yeah.
But that's the whole idea. But I don't know is any, in fact, any idea about that, Professor Peng, please.
Yeah. So like Professor Zahami, let me try to recap your question. Like let me know if my understanding is correct. So you're asking like whether there are any existing work to study the metabolite inside the tumor, inside the cancer cell, to study their association with cancer outcome from aging, immune cell aging, right?
Exactly. Yeah, that's true.
Yeah. So like to my knowledge, I am only aware of some computational work to study the metabolite flux still based on gene expression. Like for example, those methods basically just look at some key metabolomic pathway to look at the average gene expression level to see whether some metabolites are being synthesized or not. But in terms of systematic profiling of metabolite levels and their association with cancer outcomes, I'm not really aware of any such studies for a few reasons. First of all, in order to detect metabolite, you have to run mass spectrometry.
Yes.
However, for a mass spec experiment, it's very challenging. Let's see the secreted metabolite in the intercellular space as an example. Metabolites are very unstable. They are really transient signal. Like if you study them in the intercellular space, it's very hard to capture their transient level. After sample preparation, you need to use organic solvent to extract lipid, for example, to dissect them. All of this process will cause severe loss of lots of metabolite. And meanwhile, the sensitivity of mass spectrometry depends. Sometimes for some metabolite, you even cannot determine their structure. What the mass spec tells you is only a spectrum. But how do you convert this spectrum to the structure? There's no guarantee. Gene expression is simple. Sequencing, you get it. But for metabolite, let's take like a glycosylation modification as an example. All of the sugar modules could be connected in really a crazy amount of different topologies, right? Or lipid.
Yeah, that's true.
Yeah, phospholipid. There are 3 locations, and different fatty acids connect to different locations. But on mass spec, they may look at exactly the same, or it's impossible for you to differentiate them. Which means from the mass spec spectrum to metabolite structure, there's a huge gap. There's no guarantee that the mass spec spectrum will give you the structure. I mean, of course, some people can argue, I can do targeted mass spec. I synthesize the lipid as the reference standard and then run a targeted mass spec. However, not all lipid or metabolite can be synthesized. In short, our current mass spec procedure cannot tell us the metabolite abundance for most of metabolite. Therefore, I think that you are talking about a really essential frontier. I think that's probably the field with the most unknown, exciting discovery to be made. However, the current mass spec is a bottleneck. We won't be able to really reliably identify the metabolite topologies, unfortunately.
Thank you. That's very nice. Thank you very much, Professor Peng. I really enjoyed it. Yeah.
Thank you, Professor. And I think in the chat there are 2 questions here, Professor. The last one we will take. We have a head of software engineering department professor here, Ferdi Hoca, and he already said that thank you for your knowledge sharing and presentation. I would like you to know that we, several data scientists, are ready for helping studies research on big data analytics, especially machine learning, artificial intelligence, and metaverse application. His message is here. And another question from our assistant professor is related with the amount of data generated in cancer research. She said that it's growing exponentially, but translating the data into actionable insights for clinical practice remains challenging. So what are the biggest hurdles in bridging the gap between big data analytics and clinical application? So, and how do you think interdisciplinary collaboration can address these challenges? Another question.
These are all fantastic questions. Maybe let me answer the first question from Dr. Turk. What is the biggest gap between big data analytics and data and clinical applications? Several weeks ago, my colleagues forwarded me a review paper called "All Models Are Wrong, but Yours Are Useless" it's a very interesting review article talking about these questions. First of all, the data are really growing so fast. Collecting them, curating them is a big hurdle. No one will really want to do this. It's really very tedious, but somehow someone has to do this. But even after you have a really reliable computational model, who will do the clinical trial, right? For example, you create something to predict clinical outcome based on your computational model. But eventually, you still need to find a clinician who are willing to do a clinical trial for you. Or, for example, we design a small molecule or immunotherapeutics. We show this work fantastically on mouse. But who will do the clinical trial? That's always a huge hurdle. For example, in our 2022 paper, we show that the FIBP knockout will significantly enhance the adoptive cell transfer therapy efficacy. We use mouse models. We use human donor T cell in vitro models. We have very comprehensive data, but eventually we did not find any clinicians who are willing to do the trial for us because everyone is busy, right? They have no other time to do trial for other people. So this is the huge translation hurdle is the clinical trial. How do you find this? Typically in US, I don't know what the mode in Turkey. In US, there are two common modes. First of all, you are MD PhD, which means you finish both medical training and research training. Then in many universities, the MD PhD, they have both a clinical team and a medical and a basic research team. So they can easily translate their finding in their basic research lab to the clinical side because they are doctors by themselves. They can just write a protocol. For example, some of my NCI colleagues who are MD PhD, they find knockout some genes can significantly enhance T cell killing. Then they can immediately find some patient to take a trial. So unfortunately, I'm only PhD. I don't have a MD degree. So this is a huge hurdle for PhDs, which means for people who are interested in doing translational research, you better go to medical school. Being a pure basic scientist will create a big trouble in the later translational stage. And the second mode is for many PhD scientists in USA is that they find venture capital to raise money, and then they use the money to pay MD or clinician to do the clinical trial for them. So for example, my previous office mate, when I was a postdoc, he is a faculty at UPenn right now. He published a paper, found something very interesting, and then he found the venture capital raises $30 million US, and then he paid the clinician to run the clinical trial. This is the most common tracks for basic scientists to translate your research findings into clinical trials through the venture capital. But unfortunately for me, I work at the NCI. NCI is the US federal government. We are not allowed to use any venture capital money. But for people at some private or public universities, that is allowed. So though in short, I don't know what is the current situation in Turkey, whether this is also the common mode. But at least from what I see in US, and also from what I heard from my friends in China, either go to medical school by yourself, then you can do whatever you want. It's very convenient because you can just write a clinical trial protocol by yourself and then do it. If you don't go to medical school, just go for venture capitals you persuade them to pay the clinicians to run the trial for you. Hopefully this answers your question. But if you have some input, especially some mode from Turkey, welcome to input here. The second question, Arta? Yeah.
Yes, yes, you can continue.
Yeah. And the second question, several data science are writing for helping studies research on big data analytics and AI machine learning metaverse applications. So Arta, do you mind to explain the question for Ferdy a little bit more? Like Ferdy, what's your question?
We have now another question. I think you read the same one. The other one is related with another student that is not here, but they sent question to us. Given the rapid advancement in artificial intelligence and machine learning, what are the key innovations you foresee in artificial intelligence-driven cancer research over the 5 to 10 years, she said or he said? How do you see artificial intelligence enhancing the accuracy of precision medicine in predicting patient responses to treatment? The last one, I think it's, yeah.
So this is probably an important question everyone are thinking in their mind. By the way, like my view might be really different from the current popular view. Just my personal view, it may be wrong. Just take it as 2 cents, okay? So in my opinion, in the next 5 to 10 years, the most important innovation of AI is not predict clinical response, it's not design new drugs, because it's far away from there yet. From the data I showed in the first part and from also many other publications nowadays show AI is really far away from there yet. But what AI will bring true difference is some low-level applications. By low-level applications, I mean some applications that our human beings really know how to do it, but are so tedious to do it. For example, like cell segmentation, right? Like when you have a medical image, especially from multiplex immunofluorescence, you want to analyze it. The first step is I do cell segmentations, but it's too tedious for human beings to do it one by one. But then human beings can really do it well. We can generate lots of high quality human label data to train AI, which means I think in the next 5 to 10 years, AI will become really reliable for this kind of low level applications, like cell segmentation, like tissue segmentation. Those are not directly doing the clinical prediction, but these are helping human or clinicians or basic scientists to save you more time for some more meaningful task. So which means in the next 5 to 10 years, in my view, AI will definitely become a very useful helpers. For example, just like a grammar checking in Microsoft Word, something like that, or automatic like a suggestion when you are writing an article using software Grammarly, it will become really good helper for scientists, for clinicians, but it will not directly do the clinical decisions. So this is my view. Like focus on some low-level applications that our human definitely know how to do it well, and focus on that. But for really high-level clinical predictions, that's going to be really hard. Hope this answers your question.
Yeah, they thank to us about that. And if it's okay for you, can we take the last one, the last question from Professor Özlem Cansever? And she said that, dear professor, thank you very much for taking the time to talk about your valuable findings. I'm reaching the relationship between low-grade brain tumors and cognition. In this regard, I'm curious about your opinion on the following issues. Can an artificial intelligence model targeting synaptic plasticity or neuroinflammation processes be developed to model the effects of low-grade brain tumors on cognitive functions? What methods do you recommend for integrating biological data with data on cognitive functions?
Yeah, well, like Dr. Cansever, hope I pronounce your name correct. this will be a hard question for me. You know, like I almost know nothing about like cognitive functions or low-grade brain tumors. Eventually, for any model to be reliable in any applications, we need quite some domain knowledge. Like so for like modeling cognitive functions, like you know, eventually you have to find some type of data that you can generate on high throughput and can reliably quantify the cognitive functions. But because I'm not an expert on this field, you know, I cannot really give you any content answers. Sorry about this.
Yes, it was a little bit different from the presentation maybe, but they are curious about the knowledge that you are working on this field, especially computational biology maybe. So I think that they thank you for your answers. And of course, we would like to thank at the end for your time. Yes, it's very early there. And for being here with us, with our students, sharing your knowledge and your valuable time with us. So we would like to thank again to you and to all of the audience that are here and listening to you. So if you have something to add, we can, I think that finish here.
Yeah. So thank you so much, Arthur. Thank you so much, everyone, for giving this opportunity to communicate with everyone. Oh, Arthur, by the way, just one last comment. Maybe in the future for